Yes. So very good evening, my dear students. তো thumbnail ভালো হয়েছে তাই তো? Good. তো with a light note, let us start. অনেকদিন পর স্টার্ট হচ্ছে ক্লাসটা আজকে। Again, you know what? Link technical glitch. Let us start with this. Okay. Okay, let us start with this topic. So, the last topic, what we have done, we have completed about from this chapter called Biotechnology and its applications. What we have done is we have already completed BT cotton first, then RNA interference, then LLE, that is Humulin, genetically engineered insulin, and the last one, what we have completed is about gene therapy. We have completed gene therapy, that's for sure. So we are going to start from the next topic after gene therapy. So what is there? It's, I think we're going to talk about the biotechnological applications in medicines. Biotechnological applications in medicines. Also, we're going to talk about the transgenic animals. Though we have already completed those, let us start from the what the NCERT says about it. It was completely theoretical. Yes. Biotechnological applications in medicines. Fine. So what it says is, yes, the DNA technological process has made immense in past in area of healthcare by enabling mass productions of safe and more effective therapeutic drugs. What is therapeutic drugs? It is therapeutic, okay, therapeutic drugs. That means those drugs which normally helps in therapy. Fine. Now, further, the recombinant therapeutics do not induce unwanted immunological responses, as is common in case of similar products isolated from non-human sources. What is the meaning of that? That means normally during organ transplantation, like a kidney transplantation, you know exactly that if I have given my kidney to Deepthi. Now, Deepthi is not having the same blood group as of mine. Throughout the life, Deepthi has to take what kind of drug? Cyclosporin A. Remember that? Immunosuppressant. Now, in order her not to use this kind of medicines, she has to depend upon the biotechnological products. Because biotechnological product is going to make what? A type of therapeutics will be can be made by biotechnological process, which is going to be helping in these kind of cases where there is a need of what kind of drugs? Immunosuppressive drugs and all. Clear? At present, about 30 recombinant therapeutics have been approved for human use the world over. In India, 12 of these are presently being used, okay, or marketed. Out of the 30, 12 of them is marketed in India right now. Genetically engineered insulin, we have already talked about that. Just to give you an idea about it, in our body, the insulin remains in the form of proinsulin. It is having A, B, and C polypeptide chain. Now, if you remove this C, which makes it from proinsulin, pro means what? Inactive insulin. So what we can do is, if we can remove the free C peptide, we can make a polypeptide chain inside the bacteria. And when we put the A polypeptide chain forming gene inside the bacteria and make only A polypeptide gene, and then we can separately form a B polypeptide gene forming DNA inside bacteria by pathological product. We can make B polypeptide chain and we can add both of them by the help of disulfide linkages, by the help of disulfide SS bond. Fine. Mind you, we have to solve some of the questions after this. Fine. Lastly, we have to solve some questions. So ultimately, what we are going to get? We are going to get the same type of insulin which is produced inside human body by the beta cells of islets of Langerhans of pancreas. Clear? Otherwise, before this biology came into play, we need to depend upon slaughtered pigs and slaughtered cattle. Okay. So this is humulin, and humulin was produced in an American company. What is the name of the company? Eli Lilly company. So Eli Lilly company, 1983. It's very, very important. American company prepared 2 DNA sequences corresponding to A and B chains of human insulin and introduced them in plasmids of E. coli to produce insulin chains. And chains A and B were produced separately, extracted and combined by creating disulfide bonds. Very, very important to form human insulin. That is called humulin. It came 2020. Insulin was first hormone. Very, very important. Is the first hormone produced by recombinant DNA technology. The production of human insulin is in bacteria by recombinant DNA is possible because genetic code is nearly universal. What is the meaning of that? Genetic code is universal. What is the meaning of that? Remember, in genetic code, the properties of genetic code, that the genetic code are degenerative, they are universal, fine, they are ambiguous or non-ambiguous, fine, they are contiguous, so universal. So, and because genetic code is universal, we can make what? We can make, say for example, say we are going to make a polypeptide chain, fine, a polypeptide chain, and say for example this A polypeptide chain is made up of methionine first, then alanine, say then valine, and say the next one is isoleucine. And we know exactly methionine is produced by, come on, the genetic code. What is the genetic code for methionine? From 5' to 3'. You know it. It's AUG. Say alanine as GUC. Say valine as GUG. And say isoleucine as CGU. Fine. So you know exactly that these are the codons which can express for these amino acid and can make this polypeptide chain. And these codons can be produced from what? The DNA, which is double stranded, 5' to 3' and 5' to 3' over here. Fine. So we know exactly this portion of the DNA can make what? A polypeptide chain. So we can cut this particular part of the DNA by the help of REN. And so that is why if you know the genetic code which can make this A polypeptide chain, we can always make it in lab. Clear? Now, gene therapy. In gene therapy, we have talked about that. A 4-year-old girl who was suffering from ADA deficiency, and which leads into SCID, a type of disease which is called severe combined immunodeficiency syndrome, wherein ADA is an enzyme which helps in making what? Which helps in making immunity in the body. So if a particular patient is suffering from ADA deficiency, what we can do is we can from a healthy person we can take the particular DNA and by the help of retrovirus we can input that particular DNA inside the retrovirus and allow the retrovirus to infect the immunocyte cells or lymphocyte cells taken from the bone marrow. What is going to happen? The retrovirus is going to inject the ADA gene inside those lymphocyte cells, and we have to culture those lymphocyte cells outside the body. And then when the number is huge, we have to again implant that inside the bone marrow. And we know that the bone marrow, they are the stem cells. They have to create this kind of cells only. But do you think it is going to be throughout the life, it is going to remain, it is going to be functional? No, because these lymphocyte cells, they have got their life. So after their life is over, they are going to die. So its effectivity is going to come down. So you have to make this on a regular basis. But it can be completely curable if you can do these changes during their embryonic stage. I think it is written over there. Last line of this gene therapy, during their embryonic stage. Yes. However, the gene isolate from the bone marrow producing ADA is introduced to the cells at early embryonic stages. It could be a permanent Clear? So we can, by the help of gene therapy, say 20 years down the line, we are going to have every other treatment done by the help of gene therapy. It's possible. Fine. I think you know about the autosomal dominant cases, autosomal recessive cases, sex-linked dominant cases. So these are all the genetic problems that we are having. So we can treat by the help of gene therapy, but it's going to take some time. So this is how it happens, it occurs. Clear? Now, molecular diagnosis, this is the part which was left. We need to start from here. Molecular diagnosis for effective treatment of disease. Early diagnosis and understanding its pathophysiology. What is the meaning of pathophysiology? Pathology and physiology is very important. Study of altered physiology of the host is called pathophysiology. Using conventional methods of diagnosis, serum and urine analysis, early detection is not possible. Think about it. Using conventional methods of diagnosis, that is taking serum, taking urine analysis, it's an age-old practice, fine. It is not going to work. The technique that serves the purpose of early diagnosis is PCR, there is rDNA technology, PCR and ELISA. ELISA we know exactly, we have studied in AIDS, fine, antigen-antibody reactions, and PCR polymerase chain reaction we already have covered in biotechnology first chapter. rDNA technology we already have done it. Presence of a pathogen that is bacteria, virus, etc. is normally suspected only when the pathogen has produced a disease symptom. By this time, the concentration of pathogen is already very high in the body. So by the time you detect the type of virus or type of pathogen which has infected your body, and when they are going to show the symptom, then only you are going to get into, you are going to go for a test. आप तो ऐसे नहीं जाओगे टेस्ट करने के लिए, and it is going to be too late, है ना? What is the famous statement, Bollywood statement, Bollywood doctor statement? अब तो दवा की नहीं. So that is the statement you are going to expect that amount of time. So it's going to be too late when you are going to see some symptoms. For that, what you have to go for? To avoid that situation, you have to go for early diagnosis. And you can do it only by the help of rDNA, PCR and ELISA. Clear? Great. Now, however, a very low concentration of a bacteria or virus at a time when the symptoms of the disease are not yet visible can be detected by amplifications of the nucleic acid by PCR. I think you know what is PCR means. Polymerase chain reaction. What do we do over there? We break down the DNA by denaturation, by heating it at 98 degrees Celsius. And then we go for annealing, and the last one is extinction. And if you repeat the cycle for 30 times, you can get 1 billion times amplified DNA. Over there, they tell me the name of the bacteria which is being used. Thermus aquaticus. Thermus aquaticus. It's a, what kind of bacteria is it? No, it's a thermophilic bacteria. Thermoacidophilic bacteria is an archibacteria. So they are heat resistant. PCR is now routinely used to detect HIV in suspected AIDS patients. PCR is also used to detect mutations in genes in suspected cancer patients too. PCR is a powerful technique to identify many other genetic disorders. Clear? So you are not going to wait for the symptoms to come up. You are going to start with what? You are going to start with rDNA or PCR or ELISA. Okay. Now, a single stranded DNA or RNA tagged with a radioactive molecule is called probe. I think we have talked about probe. Tell me the term. Tell me the process where we have talked about probe, radioactive probe. Gel electrophoresis. No, that is one of the part. VNTR. No, tell me VNTR. So VNTR means we have talked about all this in DNA fingerprinting. Radioactive probe. Probe is allowed to hybridize to its complementary DNA in a clone of cells followed by detection using autoradiography. The clone having the mutated gene will hence not appear on the photographic film because the probe will not have complementarity with the mutated gene. Clear? So probe is going to be the useful one. If there is some problem in the gene, probe is going to help in the detection of it. Genes of interest can be selected from a genomic library by using DNA probes. By using DNA probes. Genomic library means what? cDNA. cDNA. ELISA is based on the principles of antigen-antibody reactions. Antigen-antibody reactions. So what is antigen? Foreign body. And if you have got that antibody in your body, and if there is an antigen coming in, it is going to get agglutinized. The process is called as agglutination. So you can easily understand by the process of agglutination that you have got a problem in your body. Fine. And that is called ELISA. So what is the basis of ELISA? The basis of ELISA is based on antigen-antibody interactions. Infection by pathogen can be detected by the presence of antigens, that is proteins, glycoproteins, or by detecting the antibodies synthesized against the pathogen. अगर आपके बॉडी में कोई एंटीजन आ गया, if you have got antigen in your body, foreign body, so they have got their own protein formation in the body. They will produce some protein in your body, no? And the presence of those protein is going to be helpful for you to understand that something has happened in their body. Their body contains glycoproteins, or by detecting the antibodies, your body has started making antibodies against those antigens. क्यों बनाना start कर दिया antibody? In your nation, India, if suddenly, army troops have started coming down to your place, I mean, something is cooking over here. Something is cooking. Got the point? So that is it. So, yes. Transgenic animals and plants. Transgenic elements and plants. We have taught many of them. Animals that have, the transgenic animals are what? Those animals or plants where a foreign DNA has been inserted. Name some, Rajanna. Name some. BT cotton. BT is a bacteria plant. Cotton is a plant. So it's a transgenic plant. Tell me some more. Agrobacterium. Okay, that is also. That tobacco plant. Yeah. BT brinjal. Name some more. Golden rice. Name some more. Flower saver tomato. Flower saver tomato. And some, we have talked about EPSP, enol pyruvate shikimate phosphate, weedicides, resistant plants. Yes. So these are what? Transgenic plants and transgenic animals we have used. Rosie the cow. Okay, we are going to talk about that. Animals that have their DNA manipulated to possess and express an extra foreign gene are known as transgenic animals. Uses of transgenic animals, normal physiology and development. Transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal functions of the body and its development. Example, study of complex factors involved in growth, such as insulin-like growth factor, by introducing genes from other species that alter the formation of this factor and studying the biological effects. Have you got this idea? Transgenic animals can be specifically designed to allow the study of how genes are regulated. And how, I think you know what is called gene regulation. We have already talked about lacoperon, tryptophan, how the genes switch on and switch off. Presence of lactose is going to make it switch on or switch off. Switch on. And that's why they are called inducible operon. And what about tryptophan operon we have talked about? The presence of tryptophan switches the operating unit. That's why they are called repressible operon. Do you remember that? Okay. So, now, study of diseases. Many transgenic animals are designed to the understanding of how genes contribute to the development of disease. These are specially made to serve as models for human diseases so that investigation of new treatments for diseases is made possible. Transgenic models exist for many human diseases such as cancer. See, transgenic models exist for many human diseases such as cancer, cystic fibrosis. Come on, tell me. Cystic fibrosis is an example of. Choose out of these four. It's an example of autosomal dominant, autosomal recessive, sex-linked dominant, and sex-linked recessive. Autosomal dominant. Cystic fibrosis. Rheumatoid arthritis. Rheumatoid arthritis. Tell me, in the chapter human disease, we have talked about, is an example of some disease. What is that disease called?
Disease.
Autoimmune disease. Autoimmune disease. Rheumatoid arthritis is an example of autoimmune disease. Tell me another one. Crohn's disease. Very good. Crohn's disease. Another one. Japan. Hashimoto disease. Very good. And Alzheimer's. Alzheimer's. Okay. Biological products. Medicines required to treat certain human disease can contain biological products, but such products are often expensive to make. Transgenic animals that produce useful biological products can be created by the introduction of a portion of the DNA or genes which codes for particular products such as human protein. Very, very important for your NEET exam and your for board exams. Fine. A human protein named alpha-1 antitrypsin. Alpha-1 antitrypsin used to treat emphysema of lungs. Emphysema is caused due to. In class 11, name the chapter where you have studied breathing and exchange of gas. The last line about the respiratory troubles, respiratory problems, emphysema of lungs is caused due to the tobacco smoking, cigarette smoking. Okay, emphysema. So we can cure this problem by the help of a human protein gene introduction, and that is which can create alpha-1 antitrypsin is used to treat emphysema. So it's a very useful product. Which one? The protein alpha-1 antitrypsin. Alpha-1 antitrypsin. Similar attempts are being made for treatment of PKU, phenylketonuria. Tell me, it is an example of autosomal recessive. Very good. Autosomal recessive. Name some few examples of autosomal recessive. Thalassemia.
Color blindness.
No. Sex-linked recessive. Albinism. Albinism. Some more. Sickle cell anemia. Sickle cell anemia. Okay. In 1997, similar attempts are being made for treatment of PKU and cystic fibrosis. In 1997, star mark. In 1997, the first transgenic cow, क्या नाम है? Rosie, produced human protein and its milk, 2.4 gram per liter. It's very, very important. 2.4 gram per liter. You have to remember this. Mug it up. 2.4 gram per liter. What is it? Protein. 2.4 gram per liter is a huge amount of protein. The milk contained the human alpha-lactalbumin. So the human milk which contains alpha-lactalbumin, this gene is being introduced inside the cow. And this cow is going to give the same type of alpha-lactalbumin. Those mothers having problem with, lactation, they are going to, make a cue. They are going to depend upon Rosie for this, and was nutritionally a more balanced product for human babies than natural cow milk. Clear? Tell me what is so very special about milk for a baby. What kind of food is milk? It contains all the 10 essential amino acids. It contains all the 10 essential amino acids. Name some. Name the 10 essential amino acids. Matt will fill. Methionine, arginine, threonine, tyrosine, valine, isoleucine, leucine, phenylalanine, histidine, lysine. Vaccine safety. Vaccine safety. Transgenic mice are being developed for use in testing the safety of vaccines before they are used on humans. So before a vaccine is being used on human body, it is being used in the transgenic mice. Now, what is the meaning of what is the specialty about that mice? The mice is going to become susceptible or vulnerable towards that particular vaccination. If it becomes more susceptible, that means it is going to work. Got the point? Transgenic mice are being used to test the safety of the polio vaccine. If successful and found to reliable, they could replace the use of monkeys. So after using this, transgenic mice, they would use on transgenic monkeys for safety of batches of the vaccine. Clear? Why monkeys? Who will tell me? Why not straight away from mice to human? Why they are not going to use straight away from mice to human? Why they need to check it with monkeys? Genetically, monkeys are much more similar to you. You have got some monkeys still there? Yes. Chemical safety testing. The procedure is same as that used for testing toxicity of drugs. Transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. They are exposed to the toxic substances that the effect studied. Toxicity testing in such animals will allow us to obtain results in less time. That is why we need to use transgenic animals, because they are going to become much more vulnerable towards that particular chemical testing. Think about ethical issues. Very, very important. The Indian government has set up organizations such as GESE, Genetic Engineering Approval Committee, which will make decisions regarding the validity of GM research and the safety of introducing GM organisms for public services. So you cannot make, you being a researcher, you cannot make a genetical product and just you can market it down. It will be checked by GESE, Genetic Engineering Approval Committee, before you are being allowed to market it. It's not very easy for you to just market it. See, it's a very, very important question, okay? Biopiracy, the use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment. What is the meaning of biopiracy? Say, for example, a chemical. I think all of you in your household, you have got Borlin. And Borlin is a product of Bengal Chemical. Now think about it, it's a patent product. That smell of Borlin, you cannot get it from any other close, products which is made by some other company as Boro Calendula, and it failed because Borolin is having a special patent product. It's a chemically it is made, and that is patented. That means if you want to buy that patent, you need to pay the compensatory amount. Got the point? What is called compensatory amount? You have to buy that. ना आपको तो वो खरीदना पड़ेगा. Clear? बायपारेसी चलता है, बहुत चलता है, बहुत चलता है. Fine. So I can give you one idea of a biodiesel concept. Biodiesel from jatropha seeds. One of a farmer, very downtrodden. Coming from a downtrodden family, a farmer's child, he made that particular biodiesel by the help of jatropha seeds. Went down to and found that it works as a fuel. Went down to a particular researcher, to a scientific researcher center. They took the particular formula, slapped the boy, and said, "You know nothing." Fine, and mind you, it has been published by those. Individual. So this is what biopiracy is. Who has get all the rewards? That particular research center or some individual. Got the idea what is called biopiracy? So this is called biopiracy. It's always there. NEET 2018, 20 and 21. Very, very important biopiracy. Rice is an important food grain, the presence of which goes back thousands of years in Asia's agricultural history. The estimated 2 lakh varieties, ज़्यादा नहीं है, कम है. 2 lakh varieties of rice in India alone. The diversity of rice in India is one of the richest in the world. Basmati rice is a distinct one. Is a distinct for its unique aroma and flavor. 27 documented varieties of basmati are grown in India. It's a documented one. I knew 27 documented one. There is a reference in basmati in ancient text, folklore and poetry as it has been grown for centuries. Basmati rice. Now, it should have some patent. It should have some patent. Yes. In 1997, an American company got patent. See. American company got patent rights on basmati rice to the US Patent and Trademark Office. Now, if any other country wants to take that particular basmati rice has to buy it from US. And it's been grown, it's the origin of what? It's the origin of our nation. Think about who has bought it. US. This allowed the company to sell a new variety of basmati in the US and abroad. Sell, huh? Sell. Without paisa or with paisa? Without paisa. Sell. So they're going to make money. Got the idea what is called patent? Got the idea what is called patent? Yes. Basmati had actually been derived from?
Indian farmers varieties.
Got the point? Indian farmers made basmati rice and now you have to buy it from US. Okay. Indian basmati was crossed with semi-dwarf varieties and claimed as an invention of or a novelty. Acha, basmati was crossed with semi-dwarf varieties and claimed as an invention of a novelty. What is semi-dwarf variety? They are semi-dwarf variety means it's not a tall variety. Semi-dwarf variety. When a particular plant is going to remain as a semi-dwarf, that means it is having much more branches. Now, if a plant becomes taller, what is going to happen with in the rice fields? If they have got taller one with the wind, with a flowing wind, they are going to post-harvest losses will be more. Pre-harvest losses will be more. Before harvesting, you're going to have the losses. So semi-dwarf variety is that. The patent extends to functional equivalents, implying that other people selling basmati rice could be restricted by the patent. See, you cannot sell off basmati rice. Only the one who are having the patent can sell the basmati rice. Several attempts have also been made to patent uses, products and processes based on Indian traditional herbal medicines, turmeric, neem. Tell me the scientific name of neem. And the scientific name of turmeric. Tell me. Do you remember that? Neem is very, very important. Azadirachta. Azadirachta. And the rest you know, I think. Dirachta indica. Azadirachta indica. Tell me what. Turmeric. Curcuma longa. Curcuma longa. Curcuma longa. Curcuma longa. Okay, chalo. The Indian Parliament has recently cleared the Second Amendment of the Indian Patents Bill that takes such issues into consideration, including patent terms, emergency provisions, and research and development initiative. Mind you, these terms are very important. You can get in your options. Okay, the transfer of genetic material from one bacterium to another through the mediation of a vector like virus is called as transduction. I think you know that. Can you tell me the tell me the discoverer.
Zinder and
Liederberg. Zinder and Liederberg. So first it never came as Zinder, it came as ginger. Tell me the scientific name of ginger. It starts with Gingiber officinale. Gingiber officinale. The scientific name of ginger. Okay. Gingiber officinale. Okay. So if you have been able to tell me about transduction, tell me about transformation.
Changing in DNA.
Now what is transformation? Who discovered it? Tatum. And what about conjugation?
Ginger.
No, again ginger. Conjugation by Liederberg and Tatum. And what about this? Transformation. Frederick Griffith. Availability of the thermostable DNA polymerase has popularized the PCR. Availability of thermostable DNA polymerase has popularized the PCR. Come on, tell me why? Thermostable. Availability of thermostable DNA polymerase. Name the bacteria which makes it thermostable. Thermus aquaticus. Very good. Extensions of primer end of the template DNA is catalyzed by Taq polymerase. It is not tagged. It should be Taq. Taq polymerase in a PCR reaction. What is Taq stands for? Thermus TAC is very, very important, huh? TAC. TAC. TAC polymerase means Thermus aquaticus. Thermus aquaticus. Thermus aquaticus. Okay. Carimolis. Come on. We have talked about Carimolis PCR. Was awarded the Nobel Prize for the development of PCR technique. Carimolis. Last day we talked about. Okay. Nobel Prize for the development of PCR technique. That's all about this chapter, I think. So let us start with some of the questions.
হয়ে গেছে
হয়ে গেছে
3 দিন ধরে পড়েছি এক কোশ্চেন
এই কোশ্চেন নো নো হয়নি হয়নি নো 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 নো
সিকিউ হয়ে গেছে আমার দুবার করা হয়ে গেছে দুবার করা হয়ে গেছে
এইটা এইটা হয়নি আই এম টেলিং ইউ কাম অন ট্রাই
ওই তো অপশন করেছি বি হবে তো সি এর জেনেটিক্যালি
GM Gordon B4 B4
অপশন ডি
B4
অপশন ডি পুরো
A2 জেডি ইঞ্জিনিয়ার ইনসুলিন ইকোলাই
এটা হচ্ছে জিন থেরাপি
ADA gene therapy ELISA antigen antibody interaction. So answer is D.
13 কোশ্চেন
না করিনি আমরা we haven't done it. No, I'm telling you.
alpha 1 হচ্ছে হলো emphysema A3
Enzyme replacement therapy
option C
ADA
option C
antitrypsin emphysema cry1 now this two cry1AB cry1AB or cry2AB cry1AC cotton ball cry1AC cotton ball what is the answer
option C
C is the answer C is the answer
BT cotton bacillus thuringiensis
BT cotton bacillus thuringiensis option
D
BT cotton bacillus antideference serodifference PCR
detection of HIV gene therapy and
endocrine gene therapy
option D
D is the answer
আমার
is it তোদের কোশ্চেন মনে আছে
আমার কি যে দুবার
কি করে হয়?
দুবার হয়ে গেছে কোশ্চেন
কি করে হয় is it চলো do it this is the last question I think read it carefully we haven't done this
একদমই তো ঠিক
কারেক্ট হ্যাঁ একদিন ঠিক আছে
hormone insulin contain extra stretch of C peptide or extra stretch বলতে পারবো
is it extra stretch or it was in between
ও না in between আচ্ছা তাহলে অপশন 3 4 5
3 4 5 are correct insulin দুটো হবে না দুটো হবে না
ডাইসাইক্লাইন বন্ড ঠিক আছে
হ্যাঁ
কম্বাইন্ড সেপারেট
হ্যাঁ হ্যাঁ তাই তো A and B polymerase chain of insulin were produced separately in E. coli. Yes, right. Extracted and combined by creating tricycle bond between them. That is correct. Okay, two is correct.
ऑप्शन 2 थोड़ी ना आंसर
मन आपको ज्यादा आंसर चाहिए मतलब टू सिंपल टू cannot be the answer.
insulin used for treating diabetes was extracted from cattle and pigs TPS
Okay, insulin used for treating diabetes was extracted from cattle and
was বলেছে হতো ঠিক আছে গ্রামাটিক্যালি ঠিক আছে
what was slaughtered? What about slaughtered cattle and pigs?
There's two.
Okay, next, prohormone insulin needs to be processed for converting into a mature and functional hormone. Is it? Prohormone insulin needs to be processed for converting into mature and functional hormone. How come? No. Some patients develop allergic reactions to the foreign insulin. Some patients develop allergic reactions to the foreign insulin. Now, what about it?
Some people বলেছে আমার দেখলে একটু
ভাই বসেন আচ্ছা টুটা রাইট না রং টু এর সাথে কোনো আছে কেউ
ওয়ান থাকলে তো হবে না
ওয়ান হবে না তার মানে অনলি আনসার ইজ টু অনলি আনসার ইজ টু অনলি and this is going to make this kind of thought process is going to make the question papers tough hai sir tough nahi hai understand cattle and pigs and slaughtered cattle and pigs are there difference or not you have to give hit to that so when you study each line each point you have to give stress equal stress in every line that's all for now I think that is yes last one yeah chalo
leg hemoglobin
wait go for it with the following protects nitrogen inside the root nodule of very good. Leg hemoglobin. Tell me, leg hemoglobin is also called as? It's a pink colored. Is also called as oxygen?
Scavenger.
Good. Oxygen scavenger. Nitrogens can only work in absence of oxygen. Sure. Next.
Human protein, alpha and fatty acids.
Yay! Cow. Cow. Lateral domain.
Human increase to
Human is lact means lactose, milk protein. That's it, I think. That's all about complete biotechnology, Babu. So we have been able to complete full biotechnology and its applications. Fine. Try solving questions in your home. Fine. Sure. That's it from me. Sure.